Hello, my name is Adarsh Kanappan, and today I will be presenting my video on analyzing the protein secretome of skeletal muscle fibroblasts in response to the different cytokines produced by exercise. Now, before I show my data collection, I'd like to go over the methodology a little bit. So first, skeletal muscle fibroblasts were cultured with fibroblast growth factor and bovine serum, and then once they reached 80% confluency, the skeletal muscle fibroblasts were then put into 6 well plates, each containing a different cytokine environment to produce proteins in that skeletal muscle fibroblast. The different ones were IL-6, IL-10, TNF-alpha, IL-1 beta, serum starved with no puromycin, which was our control, and serum starved without puromycin, which was kind of our control, but that was not used for the control. And we did serum starved without puromycin to see if there would be a significant difference between that and serum starved with puromycin, to see if the puromycin pulse that we are later going to perform affected the secretome of proteins. So these proteins were then exposed to these cytokine environments for 48 hours, and then a Western blot was run with the product from each protein. 3 trials were done for each one of these, so in total 18 trials were done, 3 trials for 6 independent variable levels each. And then 48 hours after, a Western blot was performed, and then the protein was stained with Kumasi blue. And that Kumasi blue protein was then counted through a Lowry. Then once that was done, each one of the independent variable levels were then put under a puromycin pulse. So basically, we exposed each different independent variable level, except obviously serum starved without puromycin, to see how much protein would be made within a 30-minute time span. So in the puromycin pulse, for those 30 minutes, what happens is these proteins, these polypeptides called puromycin, will go and incorporate themselves into the polypeptide chain of each producing protein. So we know that each protein that has puromycin on it was made within those 30 minutes, and then we took the amount of protein that was made in those 30 minutes and compared it to all of the other independent variable levels. So the video I will be showing you is imaging of the puromycin protein of the Western blot taken from the puromycin pulse. So what will happen is I will take the gel and I will image that gel, and then we will get a idea of how much protein density was produced for each was secreted for each level of the independent variable for trial N3, the third trial. And then the resulting picture will be put through the Bio-Rad image lab, which will approximate the amount of protein based on the density of the blue band of the Western blot. And usually Western blots with blue bands that are higher than everything else have more protein. And the image software took those bands and turned them into numbers, which I then used to draw conclusions and results.
change the exposure to 150.
And then this is me taking the image.
Yep.
And then this is the image after the timer was done. And in this video, I take the image that we produced in the last clip, and I try and put it through the Bio-Rad imaging software to get a general reading on how much total protein, or just total factor, was detected by the Western blot. So what I do here is I split it up into lanes based on the rectangles that the imaging software provides. So I split it up into the 7 lanes, 6 including the anchor. And then what I do is I take those lanes and I analyze how much the Bio-Rad imaging software picks up on, how much protein, how many components that the software picks up on. Okay, what do I do here?
Lane statistics at the bottom.
Okay. Lane statistics.
There you go.
And then once the raw protein was picked up on, so how much total protein the software found, we then divided that by the amount of protein found in the anchor. And this was done to kind of consolidate our results and produce more easily readable numbers. So that was adjusted to the anchor, normalized to the anchor is what they called it. And the normalized protein to the anchor was the protein that was used for analysis. I would like to thank Ms. Madison Taylor and the Hill Lab Cardiometabolic Laboratory for allowing me to both video myself and conduct the experiment at their facilities. And I would especially like to thank Miss Madison Taylor for all she's done for me. She was the one taking the videos and guiding me through what I had to do, not just during the videos, but throughout my entire experiment. She taught me how to perform the Lowry assay, the Western blot, how to scrape for lysates, and none of this could have been possible without her.